Hello friends, in today's lecture we are going to talk about the contributions of Robert Koch and some other scientists in the field of microbiology. Robert Koch was born in 1843 and was a German physician by profession. He is considered to be one of the founders of bacteriology for his various contributions in this field. As the district medical officer, his primary concern was the prevalent occurrence of anthrax among farm animals in his area. After converting his four-room home into his own medical laboratory, stocked only with a single microscope, Koch set out to determine the cause of this virulent disease. He was working on anthrax around the same time when Pasteur was working on it in France. Let us now discuss how Koch led us to the germ theory of disease. Koch established by careful microscopy that the causative agent, a rod-shaped bacteria, was always to be found in the blood of an animal suffering from anthrax. He took a small amount of infected blood and injected it into a healthy mouse. The healthy mouse showed symptoms and succumbed to the disease. Now, he took a small amount of blood from this first infected mouse and injected it into the second mouse. The second mouse too succumbed to the disease. He now took blood from the second animal, injected it into another and obtained the same characteristic disease symptoms. He repeated this process as many as 20 times and proved that the bacteria did indeed cause anthrax. The 20th mouse died as fast as the first, and in each case, Koch could demonstrate the rod-shaped bacteria by microscopy. This picture here is one of the first original photographs of anthrax bacilli published by Koch. Note the shape of the bacteria. What are they called? Yes, they are called bacilli. Koch carried this experiment further. He found that the bacteria could also be cultivated in the nutrient fluids outside the animal body, and that even after many transfers in culture, the bacteria could still cause the disease when re-inoculated in an animal. Bacteria from the diseased animal and the bacteria in culture broth both induced the same disease symptoms upon injection. This led him to formulate four postulates. Thus, on the basis of these and other experiments, Koch formulated what we now call Koch's postulates for proving that a specific type of microorganism causes a specific disease. The Koch's postulates are: the first one, the organism must always be present in all infected animals but never in healthy ones. The second postulate states the organism must be isolated from the infected animals and grown in as a pure culture. The third postulate states the characteristic symptoms of the disease must be produced when the pure culture of the organism is inoculated into a healthy animal. And the fourth postulate states the microorganism must be re-isolated from this experimental animal and cultured again in the laboratory. This organism must be the same as the original microorganism. Using cautious postulates as a guide, investigators following Koch identified the causative organisms of many human and animal disease. Let us now discuss the efforts and strategies exercised by Koch in the development of the pure cultures. A pure culture is a culture that contains only a single type of microorganism. Koch realized the importance of pure cultures and the development of simple methods for obtaining pure cultures. The dilution method did not always give satisfactory results. Other scientists had already experimented with the use of solid media like potato, starch paste, and bread. Koch exposed potato slices to air and then incubated them. Bacterial colonies having characteristic shapes and colors did develop on the potato slices. But potato slices had several disadvantages as a solid medium. First, the surface was moist, which allowed the motile bacteria to spread freely over it. Students, see the spreading growth here. All the bacterial colonies appear to have been mixed up. The second disadvantage is the colonies were difficult to see since the substrate was opaque. And the third disadvantage was potato mainly contains starch and many bacteria may not have the enzyme amylase to break it down and hence may not serve as a good nutrient medium for many bacteria. Koch realized that it would be better to solidify the well-tried liquid nutrient medium which was in use those days. He added gelatin as a solidifying agent in 1875. But gelatin too had several disadvantages. Some of them were it is a protein which is highly susceptible to digestion and liquefaction by some bacteria which have the enzyme gelatinase. Above 28 degrees, gelatin gets liquefied and hence is not suitable for most human pathogens which grow at 37 degrees, the normal body temperature. The problem of a good solidifying agent persisted until Fanny Hess, wife of Walter Hess, suggested a solution. Walter Hess was an associate of Koch. Fanny Hess, his wife, suggested the use of agar, a polysaccharide, as a solidifying agent. She had used agar for making jellies when she was in Japan, and the consistency of jellies had increased with the use of agar. Walter Hess experimented with agar as a solidifying agent and he was successful. He immediately reported to Koch, who recognized the great value of agar as a solidifying agent in microbiological media and started using it regularly in his work and even communicated about it to the scientific world. Agar has many advantages and had the ability to overcome all the disadvantages of the previous solidifying agents. First, agar has a melting point of 100 degrees and solidifies at 45. Thus, it remains solid throughout the temperature range of bacteria. Second, it produces a stiff and transparent gel, making the visibility of colonies very easy. Students, you can see all different colored colonies on the agar plate very easily and clearly. Third, agar is a complex polysaccharide attacked by a very few bacteria and hence not liquefied easily. So useful has been the discovery of agar that even after so many years, agar enjoys to be the most popular solidifying agent in microbiological media. Unfortunately, Fanny wasn't recognized for her contribution until 1939, 5 years after her death. Today, we know that the agar we use in our labs actually originated in Fanny's kitchen. But because it was discovered in Koch's lab, he is credited with the discovery of agar as a solidifying agent. Let us now discuss the contribution of another associate of Robert Koch and his innovation. When agar was discovered, it was common practice at that time to pour the media into flat glass plate and then cover it with a bell jar while it cooled. In 1887, Richard Petrie, an associate of Koch, decided to modify this method by putting the media into glass dishes with sides that were 15 mm tall and a diameter of 100 mm. Students, it is the same Petri dish or Petri plate that we use in our microbiology labs and whose original design has not changed up to this day, except that the disposable plastic plates are also available now. The greatest accomplishment of Koch in medical bacteriology was his work on tuberculosis. Koch's aim was to demonstrate the causative agent of tuberculosis. He isolated the causative agent tubercle bacillus. He realized that the mere identification of the organism associated with the disease was not enough. In order to prove that it was the specific cause of tuberculosis, he needed to culture the organism. The bacteria, however, could not be grown on routine culture medium. He succeeded in isolating and growing the organism using coagulated blood agar. Koch announced his discovery of the tubercle bacillus in the famous lecture given in Berlin in 1882. Students, let me tell you that there was a fierce rivalry between France, led by Louis Pasteur, and Germany, led by Robert Koch, to prove their supremacy in the field of science. Hence, at the 10th International Medical Conference in Berlin, Koch felt forced to announce good news that he had found a cure for tuberculosis called tuberculin. Countless patients and doctors traveled to Berlin to obtain the remedy. Tuberculin, however, proved to be ineffective as a therapeutic agent. Koch didn't want to admit his failure and so administered tuberculin to hundreds of people who developed tuberculosis and died. As enthusiasm for the putative remedy waned, Koch's reputation declined. In the midst of public scandal, Koch fled Germany for Africa. However, the word on the scandal never reached the entire populace because communication was not so quick in those days. Koch had other triumphs in microbiology besides discovery of causative agent of anthrax and tuberculosis. These included the discovery and isolation of causative agent of cholera, Vibrio cholerae. In 1896, Koch later went to South Africa to study the origin of rinderpest and although he did not identify the cause of this disease, he succeeded in limiting the outbreak of it by injection of bile taken from gall bladders of infected animals into healthy farm stock. This was followed by his work in India and Africa on malaria, blackwater fever, sarra of cattle and horses, and plague, and the publication of his observations on these diseases in 1900 in 1898. Soon after his return to Germany, he went to Italy and the tropics, where he did useful work on the etiology of the different forms of malaria and their control with quinine. Thereafter, Koch continued his experimental work on bacteriology and serology. In 1905, Koch received the Nobel Prize in Physiology and Medicine for his work in tuberculosis. Koch was duly honored by his own country. He became Professor of Hygiene and Director of Institute for Infective Diseases, which was founded for him at the University of Berlin. This institute concentrated primarily on isolation, cultivation, and characterization of the causative agents of many infectious diseases of man. Robert Koch died in 1910 and was laid to rest in a mausoleum inside the institute. Later, the institute was renamed Robert Koch Institute. Koch's studies inspired a generation of scientists. In the span of just 30 years, from 1876 to 1906, the principal bacterial pathogens of human diseases were isolated. His discovery of the anthrax bacillus had ushered in a gold age of discovery. His work of tuberculosis and cholera had awakened the world to the marvels of microbiological research, and his institute, dedicated to the study of infectious diseases, attracted scientists of extraordinary ability and achievement even today. My dear students, your syllabus mentions the contributions of only three scientists, Edward Jenner, Louis Pasteur, and Robert Koch, about whom we have already talked and discussed about. But I believe sincerely that history of microbiology is incomplete without the discussion of a few more scientists, particularly Joseph Lister, Elie Metchnikoff, Paul Ehrlich, Alexander Fleming, Martinus Beijerinck, and Sergei Winogradsky. Hence, we will quickly go through the contributions made by these genius scientists in the field of microbiology. Joseph Lister was a professor of surgery at Glasgow University. He was worried about the fact that many people survived the trauma of an operation but died afterwards due to post-surgical infections known as ward fever. In the Edinburgh hospital where Lister worked, almost half of the surgery patients died from such infections. Lister had read about Pasteur's work on how wine went bad because of microorganisms in the air. Lister wondered if the microorganisms in the air were also the cause of the infections which killed his patients after they had successfully survived surgery. Probably the open wounds made it easy for germs to get into the body. He started to clean the wounds of his patients with carbolic acid and soak the dressings in the antiseptic liquid as well. He invented a machine that misted out portions of carbolic acid into air around the operation table and equipment to kill airborne germs. He then experimented with hand washing, sterilizing instruments, and spraying carbolic acid in the theatre while operating in order to limit the infection. Note the person spraying carbolic acid in the operation theatre during surgery in the photograph. In the years from 1864 to 66, the death rate for Lister surgical patients was 45%. However, between 1867 to 70, when he introduced his new antiseptic treatment, this fell drastically to 15%. Lister was very eager to acknowledge Louis Pasteur's invaluable contribution. In a letter to Pasteur in February 1874, Lister wrote, "Thanks for having, by your brilliant researches, proved to me the truth of germ theory. You furnished me with the principle upon which alone the antiseptic system can be carried out." Today, although the antiseptics and disinfectants being used have changed, aseptic surgery is still the basis of saving millions of lives, and Joseph Lister is rightly called the father of antiseptic surgery. Elie Metchnikoff was a Russian who was one of the first scientists to study the body's defense against invading microorganisms. He served as a professor of zoology and comparative anatomy. It was in Messina, Italy, while studying the digestive organs of starfish larvae that he discovered phagocytosis. One morning in 1882, as Metchnikoff observed cells moving freely within transparent starfish larvae, he hit upon this theory. These wandering cells in the body of starfish larvae, these cells eat food, but they must also eat up microbes. Of course, the wandering cells are what protect the starfish from microbes. Our wandering cells, the white blood cells, must be what protects us from germs. To test this hypothesis, he inserted small thorns from the garden plants into the starfish larva. The following morning, he noted that the cells within the larvae were no longer moving around aimlessly, but were instead aggregated around the foreign bodies as if to drive them out. The theory of phagocytosis was thus taking shape. He theorized that under appropriate conditions, protected cells within an organism would mount an immune response by attacking and ingesting foreign matter. Metchnikoff coined the term phagocyte from the Greek words phagous meaning to eat and cyte meaning cell. But phagocytosis, the cellular basis of immunity, took a temporary backseat when German scientist Emil von Behring discovered humoral immunity in 1890. An intense debate erupted as to which theory was correct, with the French supporting Metchnikoff and the Germans von Behring. Eventually, both theories became entrenched as being fundamental to the study of immunology. Metchnikoff then moved to France in 1882, adopted it as his home, and chose to work at the Pasteur Institute in Paris. During his last 15 years, Metchnikoff became fascinated with the biology of aging and death. He believed that the aging body was gradually poisoned by waste products from harmful bacteria in the intestines, which could be neutralized by lactobacilli in yogurt and sour milk, and strongly advocated regular ingestion of lactobacilli. Thus, he was pivotal in starting the relatively modern discipline of probiotics. Metchnikoff received the Nobel Prize in Medicine in the year 1908 for his work on cellular immunity along with Paul Ehrlich. Talking about Paul Ehrlich, he was a German medical scientist known for his pioneering work in hematology, immunology, and chemotherapy, and for his discovery of the first effective treatment for syphilis. He was obsessed and fascinated with the affinity of stains and dyes for tissues and the underlying chemistry of the reaction that he pursued his doctoral research on contributions to the theory and practice of histological staining. When Koch had announced the discovery of the tubercle bacillus in 1882, Ehrlich lost no time in developing an effective stain to demonstrate the tubercle bacillus and establish its acid fastness. Ehrlich continued to experiment with cellular staining. Ehrlich showed that all the dyes used could be classified as being basic, acid, or neutral, and also differentiated the numerous types of blood cells of the body. His work on staining of granules in blood cells laid the foundations for the field of hematology. The selective actions of these dyes on different types of cells suggested to Ehrlich that chemical reactions were occurring in cells and that these reactions formed the basis of cellular processes. From this idea, he reasoned that chemical agents could be used to heal diseased cells or to destroy infectious agents. He prophesied that chemists in their laboratories would soon be able to produce substances that would primarily seek out specific disease causing agents. He called these substances magic bullets. Little did he realize that it would be a 17-year-long journey before he would develop salvarsan, the first effective chemotherapeutic agent against Treponema pallidum, the causative agent of syphilis. Salvarsan was the 606th chemical agent he tried and was extraordinarily effective and harmless despite its large arsenic content. It seemed as if the magic bullet had been found. Ehrlich did further important work on immunology and showed that the toxin-antitoxin reaction is accelerated by heat and retarded by cold, and that the content of antitoxin in the antitoxic sera varied so much that it was necessary to establish a standard by which their antitoxin content could be exactly measured. This work and his other immunological studies led Ehrlich to formulate his famous side chain theory of immunity. He received, jointly with Elie Metchnikoff, the Nobel Prize for Physiology and Medicine in 1908 for the discovery of salvarsan. Let us now talk about Alexander Fleming. Scottish biologist Alexander Fleming is widely regarded for his 1928 discovery of penicillin, a drug that is used to kill harmful bacteria. The discovery of penicillin has often been described as a miracle drug, and that is exactly what it was. Do you know the story of penicillin? Well, after a month-long vacation, Fleming was examining a culture of Staphylococcus aureus, a pathogenic bacterium on which he was doing his research, when he noticed that it had become contaminated by a mold. Although the species of the mold was unknown to Fleming then, he did observe that it was inhibiting the bacterial growth. Students, Note the zone of inhibition around the mold colony. Subsequently, Fleming isolated an extract from the mold and he named it penicillin. Although his discovery was published, there was not a great deal of attention paid to his paper. Also, further attempts by Fleming to produce a concentrated extract of penicillin failed, and he himself was unable to prove that it had any therapeutic value and later doubted it himself. That Alexander Fleming discovered penicillin by chance is actually a myth. In fact, before discovering penicillin in 1921, Fleming discovered lysozyme and found that bacteria could be lysed and killed by it. Later, Dr. Howard Florey and Ernst Chain became interested in antibiotics when they came across Fleming's paper on penicillin. After a great deal of effort, enough of penicillin was produced and extracted to be successfully tested on mice, and later human trials were carried out successfully. By this time, that is, in 1941, it was now acknowledged that penicillin was indeed a worthwhile drug and could save thousands of lives. The antibiotic eventually came into use during World War II, revolutionizing battlefield medicine. Fleming had predicted that the use of penicillin would, in time, be of limited value because bacteria would eventually recombine genetically to resist the effects of penicillin. By as early as 1952, as much as 3/5 of all staphylococcal infections were penicillin resistant. Though Fleming discovered penicillin, it took Florey and Chain to make it a usable product. Hence, Fleming, Florey and Chain, all three of them, were awarded the Nobel Prize in Physiology and Medicine in 1945 for the discovery of penicillin. Martinus Beijerinck was born on March 16, 1851 in Amsterdam and was originally trained as a botanist and began his work studying the microorganisms that were present in and around plants. He soon began experiments with microbes in the soil. His greatest contribution to the field of microbiology was the development of enrichment media. Beijerinck discovered that by adding or removing certain compounds from the medium or incubating under different conditions, it was possible to favor the growth of certain microbes and prevent the growth of others. An example is Beijerinck's work with nitrogen-fixing bacteria, which are important in agriculture and in the global cycling of nitrogen. In 1888, he isolated the bacterium Bacillus radicicola, later classified as a type of rhizobium, which lives in the root nodules of leguminous plants. Beijerinck also wanted to isolate an organism capable of fixing nitrogen in the presence of air because all previous isolates fixed nitrogen only under anaerobic conditions. He succeeded in isolating a new microbe, Azotobacter crucocum, an aerobic nitrogen fixing bacteria. Similar selective culture techniques also enabled his lab to isolate sulfur reducing and sulfur oxidizing bacteria, lactobacillus species, green algae, and many other microbes. Beijerinck also played a major role in the discovery of viruses. He worked on tobacco mosaic disease And through a series of experiments, he developed the concept of filterable agents. Beijerinck believed this agent to be a fluid and he called it contagium vivum fluidum, which means contagious living fluid. Later, this liquid was renamed virus for the Latin word poison. Yet another scientist who was drawn to microbiology because he was impressed by the incomparable glitter of Pasteur's discoveries was Sergei Winogradsky. The role of microbial ecosystems in bioremediation and agriculture has renewed the interest of the microbiologists in the work of Winogradsky. Sergei Winogradsky was interested in soil bacteria, and when working with Beggiatoa, a sulfur oxidizing bacteria, he discovered that it obtained its cell's carbon from carbon dioxide. He used the term autotrophy to describe this particular property, a radical idea at that time and one not readily accepted by other microbiologists. This led Winogradsky to propose the concept of chemolithotropy, bacteria capable of growth using purely inorganic compounds of carbon and energy. He was one of the first scientists to isolate the pure cultures of nitrifying bacteria and to confirm that they carried out the separate steps of conversion of ammonia to nitrite and of nitrite to nitrate. This led directly to the concept of the biogeochemical cycles of nitrogen and sulfur in nature and emphasized the fact that these microbes were essential for the cycling of nutrients in the environment and the living world was dependent upon their activities. Working in the late decades of the 19th century, he was also a pioneer in selective enrichment techniques for the isolation of bacteria. Yet another technique employed by Winogradsky to isolate organisms from nature was a miniature model pond cross section, which has since been called a Winogradsky column. Sergei Winogradsky was also one of the first scientists to study biofilms. He promoted the idea of viewing the soil not as a mass of dead organic and inorganic debris, but as a living environment, as a collective entity that possessed the characteristic functions of a living organism. He viewed the soil as an entity that respired, transformed organic and inorganic molecules, and kept the components of the soil in a dynamically healthy balance, and hence is rightly called the founder of soil microbiology.